Assalamualaikum, I bid to Dr. Dayang. My name is Abdul Halim Abdul Rahman, and we are from Subgroup 3. I'm going to present about a journal reflection, assessment of waste treatment and energy recovery from dairy industrial waste by anaerobic process. So for the introduction, the problem statement state that the process of discarding of huge amount of organic waste without a proper treatment can cause environmental pollution. And some conventional methods need a lot of energy and high costs, and also the process only certain amount of biological slime that need to be disposed eventually. And for the biological slime may cause a huge problem at the landfilling sites. And the food processing industry, which is one of it is a dairy, known to be contribute in major production of methane gas. So the objective of this journal is to assess the anaerobic digestion of dairy industrial waste, such as sludge and effluent, and to determine the removal of organic load and energy generation from the system. So the solution approach that they have suggested are anaerobic digestion, which is known to be a fine process that's suitable for many types of organic waste treatment, and also recognized for its economical and removal steps of biodegradable organic substances from wastewater, and also known to be more stable process that able to maintain the both environmental and energy, and also converts over half of the effluent COD, which is chemical oxygen demand, into biogas. And also the parameters consideration for this designing anaerobic digestion are the pH, the temperature, the retention time, organic loading rate, carbon to nitrogen ratio, and also mixing.
For materials and methods, the wastewater and sludge were collected from Dairy Industry Private Limited, Lucknow, whereas cattle dung was collected from the nearby area of the university campus of BBA University, Lucknow, India. And then the collected sample was stored in plastic container at 4 degrees Celsius until further use. Okay, next, move on to analytical methods. Chemical oxygen demand, biochemical oxygen demand, total solid, total organic carbon, and pH analysis were performed at the Environmental Quality Lab as described in Standard Methods of APHA 1995. Carbon is a main factor for the energy content of organic compounds. Carbon were measured as total organic carbon. And for the gas production, it was monitored every 5th day by a water displacement device attached to each digester. Next is experimental setup. The experimental study was divided into two phases, which are Phase 1 and Phase 2. Phase 1 contained pure feedstock materials and labeled as P1, P2, and P3. Meanwhile, Phase 2 has mixed compositions of pure feedstock materials with inoculum in anaerobic condition and labeled as M1, M2, M3, and M4. The experiment were carried out with identical digesters of 5 liter capacity each with liquid displacement system for biogas collection. The digester containers maintained at mesophilic temperature and were fabricated using leak-proof sealing. For both phases, the digester feed were prepared using without and with inoculum, mixing it with dairy industry waste samples. The content of digester were mixed thrice a day by stirring manually up to 10 minutes to maintain intimate contact between the microorganism and substrates. The experiment was carried out for 50 days.
Next, I will continue with the results. In this study, analysis of pre- and post-digestion period on day 1 and after 50 days of hydraulic retention time for initial and final slurries from all the digesters are shown in the table below. The percentage removal of organic loading in context of total solid, total organic carbon, biochemical oxygen demand, and chemical oxygen demand for waste treatment are also recorded. In Phase 1, 2 replicate digesters of pure feedstocks containing the same amount of anaerobic feed material were run to determine the reduction in organic pollution load, total solid, and pH variation simultaneously for biogas generation. While in Phase 2, A significant improvement was observed using inoculum with feedstock materials of phase 1 for both parameters, which are the waste treatment through percentage of removal in organic pollution load and energy recovery. Based on the results of the cumulated biogas production at 50 days of hydraulic retention time, the highest percentage reduction, which is 59.3% in organic load, was measured with P3 digester only, which consists of pure kettle dung as slurry, shown in Figure 1. At initial and final day of the experiment, total organic carbon values are also observed, where a gradual decrease in values has been measured. These results fully support the experimental data by which the lowest conversion of phase 1 study with pure feedstock slurries to biogas was not obtained at P1 and P2 digesters due to very less amount in percentage removal, which is 9.6 to 32.8% as shown in Figure 2.
Organic pollution load removal. The pH values increase at final slurries in all digesters is because microorganisms require a neutral or mildly alkaline environment to produce gas. Results show that only digester P3 can produce biogas. This is due to the high amount of percentage reduction, which is 59.3% for COD. The result also state that the digester P3 has a ratio 0.53 in BOD/COD, and the ratio between 0.3 to 0.6 indicate the possibility of biodegradation. Biogas generation. The biogas generation was observed every 5-day interval of study because there is no rapid increase was observed for all digesters for this phase. Based on the result, the reason why only digester P3 has biogas generation between day 5 to 35 is because of the easily degradable part of the organic contained in the P3 was digested. Organic pollution load removal. Based on the result, COD level is high at initial stage. Anaerobic treatment is used to reduce the organic pollution load and brings down BOD and COD from original initial. Anaerobic treatment typically takes a significant period of time to be fully effective, but it can be reduced with the addition of inoculum. According to result, digester M1 has the minimum biodegradation rate related to BOD/COD ratio, which is 0.2 and causing no production of biogas. This is because of the absence of nutrient and lack of water to enhance the microbial activity. But for digester M2, M3, and M4, the production of biogas happened because of the addition of kettle dung with dairy waste or inoculum, causing a more efficient energy producer. Biogas generation. The summaries of the biogas generation in descending order are shown in the slide. Similar to digester P3, a rapid initial production was followed by a gradually increase from day 10 to day 40. This may be due to the easily degradable part of the organic in feedstock. The result indicate that the addition of kettle dung with dairy waste as inoculum made the energy producer more efficient. Based on the 4 digesters from phase 1 and 2, it showed that both dairy industry waste product, which is sludge and influent, shows efficient activity in the presence of inoculum.
Next is conclusion. Based on this study, the parameters of dairy industrial waste such as biochemical oxygen demand, chemical oxygen demand, total solid and total organic carbon show a higher rate of percentage reduction in values after anaerobic digestion. And simultaneously, the potential for biogas production in waste was also observed. In phase 1, it can be concluded that pure dairy sludge and wastewater was not a potential source for gas generation at individual level, while in phase 2, sludge, wastewater, and kettle dung, which is act as inoculum, in combination produced potential gas production with maximum chemical oxygen demand removal efficiency comparative to pure feedstock of phase 1. And lastly is reflection. Apart from treating the wastewater, the biogas produced from the anaerobic process can be recovered. Besides, the system is comparatively easy to operate and cost-efficient in a sustainable approach. The anaerobic process may be perceived as a potential alternative as it provides a renewable source of energy and utilizes recycling potential of degradable organic portion of waste generated by numerous activities in the country. This waste management technology capable of maintaining both environmental and energy concerns because it has dual benefits such as pollution control, energy production with microbiological degradation of organic pollutants, reduction of global warming potential, and diminishment of odor.